Okay, it's time, I think, I hope anyway, to move forward in this review session. As always for my review sessions, there's nothing new here. It's all stuff that I think I either didn't do a terribly good job of explaining. I also talk really fast, so I trip over my voice. The second thing is, particularly since we'd have a guest lecture, I wanted to go over a couple of things there. No guarantees whatsoever that any of this will be on the exam, but they are things that I thought were important, and I am still writing the exam. So there may be a correlation between the two of those. Any questions just about the exam for Wednesday? You can have as long as you need. Just make sure you're here on time to start. I let people take the exams with them when they go. So that means if you get here after the first person is done and has turned in their Scantron and taken the exam, I can't let you start the exam. So just be here on time. Yeah?
This is going to seem silly, but I've never heard a teacher say this before. Do we literally have as long as we need? Like if I want to hang out here for 3 hours taking this exam?
Yeah, so the question here is, will I let people take 3, 4, 5, 6 hours? And the answer is yes, I will. But I've also plotted the scores with time, and there is absolutely zero correlation between how long you spend on the exam and how well you do. So you'll end up second-guessing yourself. And I've had lots of people come up to me in office hours afterwards and say, oh, I had that right and then I switched it. So again, it's going to depend on each one of you, but generally I would say go with your gut on most of these things and don't overthink too many things. Yes?
So I went and checked for the Scantrons on Friday.
Yeah.
I know Smith is completely out of them because I guess the biology department, there's one teacher that had each student by 9 per exam, cleaned them out.
Damn biologists!
I guess more people are switching to them and they weren't aware of that. Do you know if the bookstore still has them? And if not, then how are we supposed to get a hold of them?
They did on Friday. They had a mountain of them.
Okay, so apparently, yes, they do have them in the bookstore. They had a mountain of them. Whether they still do, I don't know. They do. So apparently the bookstore is the place to go and get those big Scantron sheets and those darn biologists. Another question up here?
I don't know if I was going to see it on the syllabus. What exactly Scantron do we need?
Oh, the big blue one, the full size.
Okay.
Any other questions, comments, worries? It's going to be on everything up through, again, the end of last lecture on Friday. I know a couple of people have to leave after the clicker questions today. If you do that, please be relatively quiet when you leave. So give those people actually some more time. You can get out your clickers now. And hopefully, I put them up a little bit higher here so people here can see them. What is the unique activity of DNA polymerase I that is not present in DNA polymerase III? 5' to 3' polymerization, 3' to 5' polymerization, 5' to 3' exonuclease, 3' to 5' exonuclease, endonuclease. Yes, that's fine. That is yours. Boom.
Thank you.
You're welcome. You can sort of see from here. Good. Just checking. Trying to move stuff up a little bit. 10, 5. I think this is actually a record for the number of clickers that we had in class. Hopefully nobody has like 5 of them that they're voting with. We shall see. So basic message here is, again, the big difference between DNA polymerase 1 that's not present in DNA polymerase 3 really has to do with what? What does DNA polymerase 1 do that is really specific to DNA polymerase 1 and only present in bacteria? Helps to remove RNA primers. And how does it do that? Because it's got a really special exonuclease activity. So it's going to be one of these two. What's the 3' to 5' exonuclease activity? That's your proofreading activity. Does DNA polymerase 3 have that? Yeah, all replicative DNA polymerases have proofreading activities. So it's not just the DNA polymerase delta has it. We'll talk about some other polymerases that don't, but for the most part, all the polymerases we've talked, DNA polymerases we've talked about so far do have proofreading activity. So the big difference here is this 5' to 3', because that's the one that chews up in front of it. What does that mean about DNA polymerase 1 that would be a real problem if it were like the polymerase that you had at a replication fork? What would be a big problem if you had 5' to 3' exonuclease activity? What if DNA polymerase 1 was really processive and stayed on the DNA a long time? It would chew up all the DNA as well as chewing up the RNA. So DNA polymerase 1 does not interact with what? What is the processivity factor? Sliding clamp. So there's no interaction between DNA polymerase 1 and the sliding clamp, because if there was, we'd have all kinds of problems because it'd be chewing up the DNA. Yeah, question?
Is it called exonuclease because it's at the end of the specific strand? That's where it's moving the primer from, as opposed to I imagined endo because I imagine in the middle of the chromosome.
Okay. Yeah, so the question here is basically endo versus exo, and that's a great question. And I guess I haven't explained it terribly well. Just the idea with an endo means you're cutting into an already extant phosphodiester chain, whereas exo is cutting in either at a 5' end or at a 3' end. So whenever there's an end that you can chew in from. So those will be your exos, and endos are going to be cutting where you have the phosphodiester backbone. Yeah?
So you keep throwing this term processivity around, and I know it has to do with, the slide clamp and everything staying on the DNA. I guess I'm just confused by that terminology. What does processivity actually have to do with, like the word just isn't quite jiving in my head.
Okay, so the question is basically, try to paraphrase here, what do I mean by processivity? And basically what I mean by processivity is that once you start doing something, you keep doing it. And so it's a process that you continue with. And so the processivity is just continuing. If you had something which is non-processive, it'll do one or maybe a couple of reactions and fall off. And so the idea with these DNA polymerases is that when you have a replicative polymerase, you don't want it to fall off. You want it to stay on as long as possible, because if it falls off, then you've got to restart again all the time. And so having the processivity factors, these sliding clamps, pCNA in eukaryotes and the beta sliding clamp in bacteria, that gives you the processivity. Does that make more sense?
So basically, it just allows the process to happen. So high processivity means that the process is sort of able to continue by whatever. It's just sort of a general term.
Yeah, the term processivity is a very general term, yes. And so the idea is, we'll talk about other processive processes that happen here. And in cell biology, people talk about processive processes. You can talk about anything where you're doing multiple rounds of some kind of reaction or binding or something like that. People talk about it as being processivity. So it's not just specific for DNA polymerases. Yeah?
So you said that only bacteria have DNA polymerase 1, right?
So I said only bacteria have DNA polymerase 1. That is correct, yes.
Talk to me about what Pol delta is doing on the eukaryotes.
So the question is, what's Pol delta doing on the other strand? We'll get to that, because it's actually one of the slides that I have in my review. So we'll get back to that a little bit later on. Okay, so, because I know some people do need to take off here. The most critical amino acid residue at the active site for type 1 topoisomerase is a cysteine, lysine, arginine, tyrosine, or serine. 10, 5. So I guess I get a good job of explaining this because, almost everyone has it right, which is nice. Yes, it's tyrosine. Why tyrosine? Because it's got an OH. What does that OH do? It pretends that it's a 3' OH, and so it hooks up to the 5' phosphate. And so that's what's happening in terms of these topoisomerases. Another really quick thing about this, a number of people have asked me, which amino acids do I expect you to know for the exam? Tyrosine would be a really good one to know. Lysine and arginine would be really good ones to know. Serine, we'll get back to a little bit later on. If anything, it's interesting that no one selected serine because serine also has a OH hanging off of it. And so, at least in theory, it could be doing something like this. Turns out it's not. So that's it. And then lysine, now if you see 2 amino acids somewhere or an amino acid, what's going on with it? Probably 9 times out of 10 it'll be lysine. So in terms of guessing, for exam, it's not a bad idea. Yeah?
By knowing, do you want us to know their abbreviations?
Oh, so abbreviation-wise, like single-letter code, no. I will always write something out like this if I'm going to be using it.
Do we also know the molecular structure?
So the molecular structures of each of these, no, I don't expect you to remember exactly how many carbons there are in any of the given side chains, but I would certainly know that lysine's got a NH2 group on it, and that tyrosine has an OH. No, I think that's a reasonable thing to expect. So again, I know a few of you need to take off. Please do so quietly. I'm going to cover now, far too quickly, everything that we've talked about so far. Yeah?
So if we had answered, say, in a test situation E, is that something that if we talk to you about our justification, we would maybe get some partial credit after an exam?
So the question is coming and talking to me about partial credit afterwards. The answer is it's extremely rare that I will do that, and if I do, I have to do it for every single person in the class. And I try and make sure that there is one best answer for every question. And basically, if you do have something like that, and okay, I've got one question that you want to come and discuss with me, I'm happy to do that. But remember, I will normalize to the highest score on whatever the exam is. So unless you've got something which is more than whatever that difference is from the high score to the 50 points, so say the high score is 40, which is normal for my exams, if you've got 11 questions that you want to talk about, you might want to come and talk to me. But I'm also very happy to talk about any questions that you have about the exam, post on discussion, etc. Happy to do that. Now, highly unlikely you'll get partial credit, but very likely that I'll answer your questions. Yeah?
Can I get clarification about the first quicker question?
Yeah.
The correct answer was the 3 to 5' exonuclease.
Correct, yes. But hang on. So it's the backing up. So sorry, it was the 5' to 3'. It was C, not D. Did I select D? I selected D on that. Thank you for picking that up. I will go back and change that.
So it goes downstream.
Pardon? So the question, so it says in the notes, it does, but so does DNA polymerase 3. So the 3' to 5' is the proofreading activity. So you always have that proofreading activity. Sorry about that. So okay. Can I say the whole thing again? Rewind, click. Okay, well actually let's go back to that question. So here. 5' to 3' and 3' to 5' is always something that gets confusing. I have to go back and look at it every single time. So that's sort of the point here. So so all polymerases are going from 5' to 3', right? That's the way all polymerases work. This second polymerase has never been found. Okay, so that's clear to people? So you're going 5' to 3' polymerizing. So you have an exonuclease. It can either back up and go from 3' to 5'. That's that backing up activity. Backing up is proofreading, because you put in the wrong nucleotide, you're taking it out. This one, the 5' to 3' exonuclease, is really bizarre. Why would a polymerase that's making DNA from 5' to 3' also chew up DNA from 5' to 3'? And the answer is because it's chewing up primers as well as the DNA. And so that's the big difference with the DNA polymerase 1, because that's different. And if I did put D in there, please somebody send me an email and say, please check, because I can go back and change all of that in the clickers. And I'll check and make sure that's the case.
Yeah, you do.
Okay, thanks. Yeah. Okay, so the question here is about 5' to 3' being downstream. We haven't really talked too much about upstream and downstream. That has much more to do with the RNA polymerase. So let's ignore that for the time being. We'll get back to it. Yeah.
Okay, so now that we've established which is which, though,
Mm-hmm.
in regard to the 3' to 5' proofreading activity that is in all DNA polymerases, is that via an exonuclease, or is that with something else?
So the question basically is proofreading an exonucleolytic activity. And basically that gets back to your question about, are you cutting in from an end? Are you cutting in the middle? So at that point, you're literally cutting in from the end because you've extended off that 3' end and put on a new nucleotide. That 3' end of the nucleotide now gets cut off. And so it's an exonuclease. It's coming in from the end of the piece that you just put on.
Okay, so it's really that DNA polymerase 1 has an extra
Exonucleolytic, yes.
Okay, but so it's still using that enzyme, just it can now go both directions for different functions.
Exonuclease in both directions, yes. Yeah.
In the notes you had 3 to 5 exonuclease in red. Is that still right, or?
Oh, that's still correct. That's the proofreading activity. So the question is, you've got two kinds of exonuclease activity for DNA polymerase 1. So does proofreading and the chopping up ahead. Yeah.
So what you would say is all polymerases have 3' to 5' exonuclease, which is proofreading. The one that's only present in DNA polymerase 1 is an extra one, which is 5' to 3' exonuclease.
I almost completely agree with that statement. And that is because there are some non-replicative DNA polymerases that don't have that proofreading activity. We haven't talked about them yet.
But
the ones we've talked about so far, the ones that you want to know for the midterm, yes.
So there's
Yeah.
a lot in there.
Sorry, last question. So the difference between DNA poly 1 and DNA poly 3 is just that 5' to 3' exonuclease. Is that the only difference between them?
So the question is that the only difference between DNA polymerase 1 and DNA polymerase 3. We talked about the processivity before. So the processivity you only get with DNA polymerase 3 because that's the one that's sitting at the replication fork and extending like mad. And so DNA polymerase 1 doesn't have that processivity. So that's another big difference between the two. Okay, so talk even faster my way through these next slides here. So a couple of these, of course, are lectures that I didn't actually give. Hopefully you listened to this one on YouTube. This has to do with microbes, mostly because most of our world is microbes. Most of our gut is microbes. There are more microbes in us than there are our own eukaryotic cells. But more importantly here is that there's a whole bunch of diversity, molecular diversity that's out there between bacteria, archaea, and eukaryotes. And if you really want to know something about molecular biology in general, you want to study really all three of these. If you want to know something about humans, it's good to study things that are relatively closely related to humans. And then just as an overview, you are here as far as that's concerned. So the other thing here, probably not as critical, this is really controversial down here. This gray zone means that people argue about it a lot. So that the first eukaryote actually came out of archaea is a pretty controversial issue there. Central dogma, basically, is what we've talked about so far. Is just right up here at the top. We've got DNA going to DNA, replication. That DNA then get copied into transcription. That's the next midterm. And then transcription, mRNA gets turned into protein. These proteins then fold up. They get modified. And that's what gives you the final phenotypes down here. It's really all those old processes that happen. The DNA, always the same. The replication is almost exactly the same, but again, you've got differences between bacteria and eukaryotes, which we just talked about. And then what genes get turned on, how those proteins get made, what modifications those proteins make, gives you what you have in terms of the final phenotype. Genomes, there's a huge variety in terms of the size of genomes. There's a log scale down here. Nucleotide pairs per haploid genome. If we go up here to humans, that's going to be about 3 times 10 to the 9, a number I do expect you to remember. Basically, we've got pretty large genomes relative to lots of other organisms, but that's mostly because it's non-protein coding. There's a lot of extra DNA that we have in our genome. I'm not going to call it junk DNA. I'm just going to call it extra DNA. On the other hand, if you look at some of the smaller genomes, particularly things like bacteria, and sorry for people over here, the E. coli genome, I don't expect you to remember this, but it's about 5 million, and archaeal genomes are even smaller than that. But some of the largest genomes are things that we probably don't think of as being terribly advanced, like the amoeba. It's got over 10 to the 11 base pairs in its haploid genome. So 100 times bigger than our genome. Has even less coding sequences in it. Why things have much larger genomes or smaller genomes is really unclear at this time. Wanted to mention, back up here, orthologs and paralogs. This is something that generally gives people issues. It gives me issues as well in terms of thinking about them. The way I like to think about orthologs and paralogs is orthos are the real ones, and the paralogs are kind of the fake ones, like the paranormal, whatever you want to think about from that. Orthodontia, getting nice straight teeth. So the orthologs are going to be the true genes of something. So alpha-tubulin, they're all alpha-tubulins. They're just in different organisms, human, fly, worm, and yeast. On the other hand, your paralog, which has come through this gene duplication that's happened, that's going to be, say, a beta-tubulin in this case. So we've got these two paralogs of alpha-tubulin in humans, beta-tubulin in humans. These are paralogs, and there's a rose because of this gene duplication event which took place, and then divergence that took place. And so some of what we have in these massive genomes is gene duplication. A lot of it, we really don't know quite how it happened. So partly because it's hard to do experiments on humans, at least ethically, we have a number of model organisms that people work with, and basically it's because they're easy to work with for a number of different reasons. E. coli, really easy to grow, relatively small genome, and the classic quote of one of the Nobel laureates, I think it was François Jacob, it could have been Jacques Monod, was if it's true for E. coli, it's true for elephants. So just thinking about molecular biology from this point of view. In the archaea, my favorite organism is Sulfolobus sulfatericus. In fact, a picture of it right here. 3 million base pair genome. Again, relatively small number of genes. If you want to know something fundamental about molecular biology, make sure it's working in all these different things. Eukaryotes, sort of the E. coli of eukaryotes is Saccharomyces cerevisiae, baker's yeast or brewer's yeast, brewing good. Also relatively small genome. One of the really important things about this, and in fact all of these other organisms here, is they're good genetic tools for studying these organisms. You can break genes and then figure out what you have broken. Also, the genomes are done for all of these organisms as well. C. elegans is really good for studying cell biology and particularly development. It's only got about 1,000 cells. We know exactly from the very first cell division that happens exactly where each of those cells comes from. Arabidopsis is, even though it seems pretty huge, 142 million base pairs, it's actually really small in terms of plant genomes, and there's good genetics for that. Drosophila has slightly larger genome than C. elegans, but people have been doing genetics on this. Again, I mentioned there's the Fly Lab. I think it's still being shown over at OMSI right now. And mice are also a really good model organism, mostly because, okay, they've got a vertebra, they're mammals, and you can do a lot more experiments with mice than you can do with humans in an ethical way. So that was it for lecture 1. How many minutes did that take? Good. And again, it's all on YouTube. You can go back and take a look at it there. Some of these other things I'll go over a little more quickly because I know that I've talked about them already. Main thing about bond energies, don't remember exactly what the numbers are, but know that covalent bonds are about 100 times stronger than most of the weak, quote-unquote, weak interactions. And even those weak interactions are considerably stronger than your average thermal motions at 37 degrees Celsius. What that means is they're not that much higher, so you're going to have them breaking and reforming all the time, and really critical in terms of biology. What are the main biological molecules we're going to be talking about? Sort of really 4 classes, but we're going to ignore 2 of them for the most part. Sugars, fatty acid, amino acids, and nucleotides. A couple things about these individual, usually people are going to talk about them as monomers because they get put together into big chains, all of our macromolecules, the proteins, the nucleic acids, etc. Amino acids have an N-terminus and a C-terminus, just like proteins have. So proteins got an N-terminus and a C-terminus. Amino acids have N-termini and C-termini. Nucleotides, not labeled on here, but the individual nucleotide, this is ATP, adenosine triphosphate. It's got 3 phosphates here. On the 5' carbon, here's your 4' carbon, 3' carbon. That's where your 3' OH is. Difference between DNA and RNA, at least in terms of the sugar, is this deoxyribose. So you're missing this 2' position, 2' OH. The triphosphates here are almost always on the 5' end, and this is what gets used in terms of putting all of the mostly macromolecules together. Sugars, the only aspect here of sugars is, yes, sugars get put together into polysaccharides. Really fascinating stuff. Talk a lot more about it in cell biology. But of course, the nucleotides are made up of these sugars as well as the bases. Again, stop me as I'm going through here if there's something you'd like me to explain in a little bit more detail. Biological processes are, when I talk about a process, it would be a pathway, multiple reactions that go together. And that's just shown here. That's going from A to E is a energetically favorable process. That energetically favorable process usually is going to be leading to breaking down of things, releasing of heat, etc. Stuff that we talked about when we talked about some of our reactions. The most biological reactions, and most of these are condensations, taking those monomers and making them into polymers, are unfavorable, and that would be like here from A to B and C to D. And this diagram would have been a lot better if you had 3 or 4 of these reactions that are in a, you get a positive delta G, so they're an energetically unfavorable reaction, but overall going here down to the bottom. And the way that you have that happen is you'll take food molecules, whatever those are, could also just be energy from sunlight, and then break them down through catabolic pathways, but somehow you've got to get this energy and use it for putting together macromolecules, the anabolic pathways. And so this would be your catabolic reaction, B to C, D to E, and the anabolic ones are going to be A to B and C to D. And the way most of that works in terms of nucleic acids and proteins is you have precursor activation, so you make a unstable, i.e. high energy intermediate in the process. And we'll take a look at that when we get to the polymerization in just a second, assuming that image shows up. Still on my computer now. Did want to talk briefly about KDs, equilibrium constants, etc. This is mostly what we're going to be talking about. It's just going to be about 2 macromolecules or macromolecule and a smaller molecule coming together. And that process is just, it's called a ligand in this case, and then the protein, so the protein interacting with the ligand. The KD, or dissociation constant, is just the product of free ligand and free protein divided by the concentration of both of them together. Also the same as the ratio of the rate constants, either binding, which would be the on rates constant, dissociation. And the main thing here, you can do this just by quickly calculating the concentration of P above, free P, is going to be the same as the concentration of bound P at the bottom. These guys are going to cancel out, and so at half bound, the free L concentration is going to be your KD. And what that tells you is it's going to be a concentration number. A very low concentration number means that almost everything is in the bound form here, because it's LP. A relatively high number concentration-wise means that it's not going to have a very tight binding. And we'll talk about DNA binding, interactions with all kinds of other things as we move on through the rest of the course. Mentioned the activation of these intermediates. This is how we have DNA polymerization. You start out with a monophosphate here down at the bottom, 1 5' phosphate. That gets 2 more phosphates added to it by ATP. And so that, the universal energy processor inside the cell, 2 hydrolysis. So it's that the gamma position, the last one of those phosphates, that gets added to the 5' ends. You need 2 ATP hydrolyses to generate, this is now your deoxynucleotide triphosphate. So this is the substrate that the DNA polymerase is going to use to add to the elongating chain. So you need 2 ATPs to make this. Then this intermediate gets added to the chain, and the way that happens is you have pyrophosphate release, which then also gets broken down to give you your irreversibility of the reaction between these two. So general chemistry here. We talked about protein structure. Main things about protein structure. The primary structure, which is that sequence of amino acids. Secondary structures, the 2 main ones are the alpha helices and the beta sheets. These are put together through the interaction of hydrogen bonds between, it's called the backbone atoms that you have in your polypeptide chain. So when we looked at their N-terminus and the C-terminus of any individual amino acid, they're going to have the side chain that comes off, but then hydrogen bond donors and acceptors, which are part of the peptide on the backbone. Those interact with each other. They can interact to form alpha helices. They can interact to form beta sheets. The big advantage of those, as far as we're concerned, what happens to the side chains where you have your variability, they stick out. So from an alpha helix, they're sticking out in a radial fashion. From a beta sheet or beta strands, they're sticking out perpendicular to that. And so that's where you're going to be able to have the specificity of interactions with other amino acids, other proteins, etc. Yes?
So one of your test questions was regarding this, and the correct answer said it was also that the interactions were 4 amino acids apart. In your previous lecture, I had written that for the alpha helix. So that's also true for the beta pleated sheet?
Okay, so the question here is basically how far apart are those individual amino acids in the primary structure, so that polypeptide chain, when they have these interactions with each other. So if you look at, when on the beta sheet here, if you look here, here's a hydrogen bond between this backbone atom here and this backbone atom here. It's the CO here and the NH over here. So this amino acid is 1, 2, 3, who knows how far away from this amino acid. So it can be an extremely long distance away in the primary sequence. On the other hand, for alpha helices, it has to be this 4 apart. Again, if you really count exactly 4, it would be this hydrogen bond donor up here. So it's more like 3.5, a lot of people will talk about as well.
It says greater than 4.
Right. So greater than 4, that would be for the beta sheets.
Question.
Yeah.
Are those the side chains of the amino
So the question is, are the side chains here held together by hydrogen bonds, or what? Basically, I'm going to paraphrase the question, what are they being held together by? It really depends on what the side chain is. And so if you've got a side chain that's, say, a lysine, then that could actually form a ionic interaction with something with a positive charge. If it happens to be a leucine, then we've got our possibility of leucine zippers. It's really hydrophobic, and so it might be interacting with other leucines. So it's going to really vary and depend on whatever those side chains are, which is, again, why you've got that advantage sticking them out. They can now interact with each other. You can get specificity that way. Okay, so, and then kind of tying back into that question, I didn't cover this, I think, very well the first time through, so I want to spend a little bit more time talking about it. Having to do with enzymes, but also protein folding. And the big picture here is that the vast majority of those amino acid side chains have nothing to do with the activity of the actual enzyme. Now, what the enzyme is doing is it's stimulating a reaction. The way it stimulates reactions is it binds to usually intermediates of one particular reaction. And this example here is binding to cyclic AMP. You don't need to know all the details of this. But we've got binding here through charge-charge interactions, hydrogen bond interactions, etc. So all those different possibilities that you've got on side chains. But to get each of those side chains in exactly the right place, you need to have all the other side chains interacting with each other. And so that's the protein folding process. Those are almost always going to be due to the weak interactions that are happening between the two. And so all those weak interactions are going to be folding the protein, getting those few amino acid side chains that you need to exactly the right place so that they can bind and stimulate the reaction, or that they can bind to DNA, or whatever it happens to be that that particular protein is doing. And so the idea here is that it's very few amino acid side chains that have to be in very precise positions in order to have this particular activity. Okay, we talked a little bit about allosteric regulation. Just wanted to go back and talk about this really briefly. The sort of big take-home picture here is, again, to sort of say, again, with that active site, it's a very small part of the protein. So a very small part of the protein. So here, for instance, the blue stuff, this is your substrate for your enzyme. It binds to a really small part of the protein. There are also other small parts of the protein, again, with very specific amino acids in them, that can bind to other molecules, other ligands that are there. That binding can change structure. And that's what the whole idea of allosteric regulation is about. Binding at one place causes a change in structure somewhere else. But again, it's not terribly surprising because, again, it's all those other amino acids that you have in your protein which are trying to get a very specific kind of structure that you need for your enzymes in the final reactivity. That you've got negative and positive is not important as far as it's concerned. We'll talk about negative and positive regulation. We talk about gene regulation later on. But in terms of structure, it's small things at one place on your protein can give very important differences in other parts of your protein as well. Another way that you can get changes in structure, other than these allosteric interactions, are the modifications that you get in signaling proteins. So there are really three kinds of signaling proteins, but there are only two on the slide. What's he talking about? So there's the ubiquitination also, which is very important in terms of signaling. We'll get to that on the next slide here. But there are really these three kinds of signaling changes and signaling molecules that are proteins that are very common molecular and cell biology. So we mentioned before, kinases, the most common protein family in the human genome, are kinases. What do kinases do? They put phosphates onto other proteins and actually very often themselves. Phosphates are great because they're big, they get a lot of charge. And so that, on the outside of a protein, is going to make a big difference in terms of what you're interacting with. These phosphates, once they've been added, they need another protein to take those phosphates off. So whenever you have a protein kinase, you also have a protein phosphatase. So it's going to be taking off that phosphate. Now, that's a big difference now between the kinase phosphatase signaling and G protein signaling. For G protein signaling, you have binding of a guanosine triphosphate. And that binding, a non-covalent binding, gives you a change in structure. The GTP, which is here, will hydrolyze. These are actually called GTPases as well as GTP binding proteins. So they're GTPases. They will hydrolyze the GTP. So in the absence of any other protein, this phosphate will be hydrolyzed from your guanosine triphosphate, and in that process will change the structure. In many cases, you do have another protein which stimulates that activity, but this protein is not required. So G proteins are different than what happens with kinases. Kinases have to have two, a kinase that turns it off, a phosphatase that turns, kinase that turns it on, a phosphatase that turns it off, whereas the G proteins often will have these other factors, but don't necessarily have to have them. The third one of these signaling molecules are the ubiquitin ligases. What does it do? It puts the ubiquitin onto another protein. How does it do this? Ubiquitin is a protein, small protein in and of itself. This needs to be activated, in fact, in a very similar kind of way to getting activation of your nucleotide triphosphates. And we talk about amino acids activation a little bit later on. So an activated intermediate, then you have this high energy intermediate here that then gets put onto an NH2 group, which comes from what? Lysines. So you always put ubiquitin onto a lysine. How do you know what protein to put an ubiquitin onto? Those are our E3 proteins. This is our specificity here. So the specificity of what protein to put your ubiquitin onto is going to be E3. E2 is the protein which will put the ubiquitin on here, and this E1 is activating the ubiquitin. But just like you have with the phosphatases and kinases, here it's a covalent change that happens to the protein that's being modified, and that has to get taken off at some point. And usually what happens in terms of ubiquitin signaling is that gets taken off by the proteasome, which chews up absolutely everything. But there are also enzymes which will cut those ubiquitins off. But they're extra enzymes. It's not something spontaneous. It's not like what you have with the G proteins. Yeah?
I remember you said the difference between G protein and the kinase and the phosphatase is there's no covalent modification.
Yeah, so the difference between kinases and G proteins is that the kinases are always going to be making a covalent bond. They make a covalent modification, whereas with G proteins, they're non-covalent interactions with the GTP. Okay, forget those boring proteins. Let's talk about exciting nucleotides. Base pairs, couple of things here. No, you may have gone back and looked at old midterms. I asked you to draw a base pair. I'm not going to ask you to draw a base pair on the midterm. Yay! It's because I don't have to grade all of them. But I do expect you to know the base pair structure, having to do with where these hydrogen bonds are, and of course that you've got 2 here and 3 here. Couple of other things about the DNA structure that are really important. Polarity, again, 5' 3', 3', 5', 5', 3' ad nauseum. I like this particular image because it does nicely show that the 3' end of your polynucleotide here is matched up with your 5' end over here. So your strands are always in opposite orientation, and that's one of, I think, the 7 or 8 problems that this nice DNA model happens to have here. Yes?
And just a little clarification about polarity. We're referring to the polar 3' end, or are we referring to the idea that they have opposite
So the question here basically is one of the great difficulties between the chemists and the biologists. Yeah. So basically the question is having to do with polarity. And I would like, when I talk about molecular biology polarity, basically just means directionality. Has absolutely nothing to do with the chemistry. So you got one orientation and another orientation. And I guess it's really unfortunate, but that's just the way the terminology is. If something like that were to come up on an exam, and you have a question about it, say, do you mean molecular biology polarity or do you mean chemical polarity? I was a chemical engineer as an undergraduate, so I totally understand it. So yeah, no, but when we're talking about the polarity here, has nothing to do with electrons and so on and so forth. So it just has to do with the directionality. So yeah, again, I'll apologize for molecular biologists now. Yeah, but again, always going to be opposite relative to each other. Watson and Crick could never have put together their model. I'm amazed they were able to put this one together without having that opposite orientations, now we'll call it, relative to the two different strands. In that process, you end up with major and minor grooves, which of course has now just disappeared here. We can look at it on this slide anyway. It doesn't really matter. Hopefully it's in your notes. But when they put together their model for what DNA looked like, it was really obvious that one side was squished and the other side was open. And that's one of the problems that this model has as well. That here we have a major groove and a minor groove. That major groove is where most of the information is here. And in fact, you can see that on this particular image because this computer doesn't like my other image here. This side, the top of each of these base pairs, is what's in the major groove. And what's on the bottom is what's in the minor groove. And hopefully you can see these two, what's called the glycosidic bond. This is the 1' position of your ribose is attached to the base. These are close to each other. They're going to be close to each other in the minor groove. That means there's a lot less space. There's also a lot less chemical interactions, different atoms that you can interact with. Anybody, say me, were to ask you on an exam how to know where the major groove is, the easiest and fastest way is to find this methyl group right here. Methyl group on thymines. They're always going to be in the major groove. That's just the way the base pairs are fitting together with each other.
You said on thymines?
Pardon?
On thymines?
Thymines, yes. So the thymines have their methyl group. None of the other ones have methyl groups, which of course you know because you've been studying your base pairs. Yeah, that of course doesn't interact in terms of making hydrogen bonds, but of course it can interact with whatever other thing is going to be interacting with the DNA. So that's the critical aspect of major and minor grooves. And again, we'll talk much more about this after the midterm when we talk about protein-DNA interactions, etc. Wanted to briefly mention one of the probably most amazing things about DNA, and that is that you can pull the two strands apart and they come back together. And you can do this ad nauseum, basically forever. And one of the things I think I did a bad job of explaining here is that's what you can see here, this melting curve. You can heat up your DNA, it'll come single-stranded, you cool it down, it will become double-stranded, and so on and so forth. And it will happen very specifically in terms of those base pairs, in terms of your sequence, as long as you do it relatively slow enough. Human genome, big take-home message here is that it's ginormous. Again, it's about 2 meters in length, any individual cell. If you're to line up all of the nucleotides right next to each other, really kind of inefficient because you've only got 4 bases, and also because there's a whole bunch of non-coding stuff in that particular genome. Human chromosome 22, one of the little ones, which has quite a few genes in it, but even in those genes, we'll take this black one here as an example, very small amounts of that, little red lines here, those are your exons, which are actually going to be coding for protein. and they're usually between 8 and 145 base pairs. So a lot of extra DNA in there. And now quite what that extra DNA is doing, we don't completely understand. We'll talk again more about that a little bit later on. Because it's so long, you got to compact it. And also, as we'll see more, we'll talk about DNA repair, and it's really susceptible to damage. You need to protect it as well. Yeah?
So you got a question over here.
Oh, sorry.
I have one as well.
Yeah.
Yeah, so I was just,
Thanks for...
getting the clarification on the cartoon on
Yep, previous one?
previously.
Yep.
So the 22-48 up at the top right there.
Mhm.
So is that saying there's 48 times 10 to the 6 nucleotide pairs within the 22 chromosomes excluding the sex chromosomes?
Okay, so sorry if I didn't explain that terribly well. Sorry, the question here. Basically, what's this 22 to 48 mean here? So what it means is, and again, I'm sorry it wasn't clear, this is human chromosome 22. So human chromosome 22, which is a relatively small chromosome, is 48 million base pairs long. So that's what that is. And 48 million, yeah, it's actually one of the smallest chromosomes. If you look at some of the karyotypes, 22 is really tiny. Yeah?
So that's what you mean when you say small chromosome
Mhm.
to the top left?
Yes.
That's what you mean by that?
When I say on a small chromosome, yeah, chromosome 22, again, if you look at all of the autosomes, that's one of the smaller ones.
The genome is huge, but that 22 is...
That one's a relatively small, but even in the relatively small one, you've still got a bunch of extra stuff, which means you've got to compact them with nucleosomes. So, among other things, just starting out with the nucleosomes. So the nucleosomes, okay, yes, they're wrapped about 147 base pairs. How do we know that? The way we know that is by purifying away your DNA bound to just the core nucleosomes, which will give you your beads on a string. And beads on a string, very, very rarely something that you're actually going to see inside a cell. It's usually once you've purified it away from a lot of other things. Once you've done that, then you can treat with nuclease, and that nuclease is going to chop up all the DNA that's not associated with proteins, that's not being protected by the nucleosomes. Then you can separate that DNA from the protein. You see that DNA is about 150 nucleotides, actually varies from species to species, which is very interesting. And then you've got 8 proteins here, which turn out to be 4 sets of dimers, H2A, H2B, H3, and H4. Yeah?
Which nuclease would you use to make the 145 or 150 base pair?
So,
And then which one would you use to dissociate the histone?
okay, so the question is, which nuclease would you use to break down the, people call it the spacer DNA between the nucleosomes? And what nuclease would you use to release the nucleosome core? So it turns out you don't use a nuclease for releasing the core. That's actually a high salt condition, which will disrupt that. The nuclease that you use here is just a DNase. It's called micrococcal nuclease, but that's not important. Just a DNase in that process. So what do you see in each of those nucleosomes? You've got, again, these 4 different core histones, but 2 each of them. So you've got 8 total which are in there. They all fold up on each other relative to this conserved 3 alpha helices, which all dimerize with each other. This is the histone fold. And then you've got all these tails, the things that are not part of the histone folds. So this is moving on into Dr. Bartlett's lecture. What happens to those histone tails? Lots and lots and lots of modifications. This is an overview of not even all of the modifications that are known to happen on these N-terminal tails. There are also C-terminal tails that also have modifications that take place to them. So mostly going to be acetylations and methylations, not at the same time. So if you acetylate, you can't methylate. If you methylate, you can't acetylate. And so this process of modification here, basically what it does, it serves as, Dr. Bartlett put down here, protein adapters. So basically, it's a landing pad for specific proteins. And so those specific proteins are called histone code readers. And basically, they have multiple domains This is one of those histone code readers that can recognize, because of the individual amino acids that have folded up in a particular place, very specific changes that have happened on the histones. So for instance, trimethylation at lysine 9 on histone H3, there's going to be a domain that interacts with that. At the same time, there'll be another domain also on the code reader that will interact with, say, phosphorylation of serine 24. And so whatever the mixture of those different modifications are, there'll be a histone code reader that will interact with that. And that interaction then will allow it to bring another protein to the DNA to do something. And usually doing something means taking the DNA apart, putting it back together, and taking it apart is usually important for getting transcription to take place. So let's look at one example of this. This is now histone H3. Again, you have modifications on all 4 of the histones. Turns out histone H3 has been one of the best studied, so we know the most about it. If you have histone H3 that has 3 methyl groups on lysine 9, this leads to gene silencing. If you have histone H3 that has 3 methyl groups on lysine 27, it leads to gene silencing. This is great, right? Methylation leads to gene silencing. Except that this guy over here has trimethylation on lysine 4 together with acetylation at lysine 9, which leads to gene expression. And so basically what that means is it's always going to be a combination of different modifications that are going to lead to different things. And so rule of thumb, yes, methylation usually leads to gene suppression, but that's only a rule of thumb. And it's biology, there are always exceptions. And the same thing is true with acetylation. Acetylation usually leads to gene expression, but not in all cases. Okay, so let's just talk about one of the examples here, and particularly important. Oh, is that a question or a stretch in the back? Okay, that's how there's a question, yes.
So these code reader proteins, is that so specific that there isn't redundancy? As in, there aren't, let's say, a backup enzyme that can fulfill the same code reading in a specific sense?
So the question basically, I'll paraphrase it here, is this a redundancy? Do you have multiple proteins that will recognize the same kind of modification and so on and so forth? People actually argue a lot about that. As far as we're concerned, for Wednesday, it's one protein recognizes one set of changes. Yeah?
When you said acetylation and methylation don't occur at the same time, did you mean like on the same lysine or same molecule, but they can occur on different lysines at the same time?
Yeah, so when I said that you can't have methylation and acetylation at the same time, that's on the same side chain. You can clearly have methylation that happens on one lysine and acetylation that happens on the other one. That was the example we had on the last slide. Okay, so these code reader complexes are very often connected to code writer complexes. What the heck does he mean by that? Basically, what that means is when you have a modification and a protein that interacts with that particular modification, we'll just say it's one for argument's sake, that protein, very often its partner is going to be a protein that makes the same modification on another nucleosome that was what it bound to in the first place. And so what this leads to is a positive feedback loop whereby you get one modification that takes place, and that modification spreads across the chromatin, right where that is. And so that's the positive feedback loop. And this is one of the ways you can get epigenetic inheritance, and that's because you have the modification leading to the same modification happening again. Yeah?
Sorry, running that. So, I don't know if you need to know this, and in which case you can ignore it, but which does what? Because when I was reading more on that, the code reader versus the code writer of what comes first, reader versus writing, and then what makes the mark, quote unquote, I got a little confused. Or do we just need to know the basics of what you just said?
So I'm trying to answer questions. So the basic question is what's on the exam? And I don't know, I'm not done yet. But sorry, but no, the basic idea here is that what's the process, and so what happens first, what happens second.
Yeah.
So the idea here, and I guess I can always claim Dr. Bartlett did a good job of explaining this, is that there's usually one event that takes place. And so here it's basically shown as some DNA binding protein that has some modification enzyme that's associated with it. And in this case, for want of a better term, we'll just say it's something which is in a trimethylate lysine 9 of histone H3. And so you trimethylate lysine 9 of a histone H3 here in chromatin. Once that happens, now you have your reader-writer complex that will pass along that signal. So it's basically a signal amplification process. So you have that one event that happens, and then it follows on after that.
Okay. So we should think of the reader-writer complex as one complex, not two separate complexes.
So in terms of the reader-writer complex, I think about it as being one complete, something that comes together, and it's usually doing both things. But you've got to outstart that. You've got to basically kickstart the whole thing. And kickstarting it, that's what you get with what's supposed to be this sort of vertical piece right here.
That's the trimethylation.
So that the first piece here, so this one, this blue piece is going to be what's going to be trimethylating your histone, or the lysine 9 in that histone. And it's brought in as part of this first one, but it's also part of this read-writer complex. Okay, because of this, and because you've got these positive feedback loops, once you start something, you have to have a way of stopping it. And that's what the barrier proteins are all about. So barrier proteins can block the progression of these read-writer complexes. Very often, that's in fact a physical block, interacting with some part of the nucleus, some part of the nuclear pore, etc. Could be something which is just physically blocking binding to the nucleosomes. Or my favorite are these anti-enzymes. And so if you have the writer complex, which is a methylase, which is putting on methyl groups, or methyltransferase, this guy could be a methylase, which is taking them back off. And so you sort of have this battle going on between the two. So the methyltransferase puts methyl groups on. This barrier protein is a methylase, which takes methyl groups off. And so that can also serve as a barrier. One of the important things here is that these guys are all bound to the DNA and all bound in specific places, specific sequences. Yeah?
Are the barrier proteins that are utilized based upon the type of reader-writer complex that's propagated down? Or is it just based upon the signaling that's actually being
Okay, so the question basically has to do with, you have specific barrier proteins depending on your specific changes which are happening. And that is definitely true for these guys, which are enzymes down here at the bottom. But for those that are not enzymes, that doesn't necessarily have to be the case. Okay, so in the last minute, we kind of talked about this already. Main thing here is you've got condensin and cohesin, which are pulling these guys together. I just wanted to cover these guys really quickly before the end of class, which is basically now. And that is just to say that these loops, which are usually separated by barrier proteins found down here at the bottom, are those that are going to have one particular chromatin state. So compacted or open, those open ones are those going to be the ones which are going to be actively transcribed. So those will be giving you your transcription, giving you the proteins, etc. We basically talked about other things already. Very last thing, people always ask me how many genes you want to know. No, you don't need to know all of these numbers. But what I would know is that there are more non-coding genes than there are coding genes that are currently present in the human genome. If you have more questions, come grab me after class. I know some people need to take off. Also, feel free to send me email. We can set up extra office hours tomorrow if people need them. Good luck on Wednesday.